to all the learners of life sciences that is watching this video. This is the third video for the series for level 7 learners that want to achieve an A symbol. In today's session, we're going to look at genetics and genetic engineering. Okay? Now, if we quickly look at the contribution of genetics in our exam paper, then we see if we look at the contribution of genetics, then we see that it is in paper 2 and that it is 30% of 45 marks of a total of 50 out of 150. So very important, if you don't understand genetics, this is the one that can cost you an A. So very important because this is very well represented in the question paper. Okay. If we look at the examination guideline, which I said to you must be your basis for studying, then we see, firstly it says that we must know the definitions of the following genetic concepts. We must know what is a gene, what is an allele, what is dominant and recessive alleles, what is monohybrid crosses, what is phenotype and genotype, what is homologous and heterozygous, what is complete dominance, incomplete dominance, codominance, multiple alleles, polygenic inheritance is out, sex-linked characteristic, karyotypes, cloning, genetic modification, and the human genome. One place where this is very, very nicely summarized is again in the book Mind the Gap. Like I remember, I said to you that Mind the Gap cover all the content. So what we listed there, the definitions, there you can see what is a gene, what is an allele, what is the difference between a gene, an allele, and a locus, what is the relationship between the two of them, what is heterozygous, what is homozygous, and what is a recessive allele. And on the next page you can see it continue with all the definitions. But we're not going to look at the definitions today, because that is what you must study. Then it said, the next thing that it says is mutations. That we must know the mutations and the effect that it have on the body. Okay, I will come back to that one just now. Then it says we must solve genetic problems and look at the genetic problems that we must solve. We must solve complete dominance, and incomplete dominance there are 3 crosses. That is where there is a homozygous cross with a homozygous, or a homozygous cross with a heterozygous, or a heterozygous cross with a heterozygous. And here, a small tip that I want to give you, but we will look at the crosses just now, is you must know the ratios of these crosses out of your head, because this will save you a lot of time in section A. Because in section A, the multiple choice, there's always what cross will lead to the following ratio. What was the parents when the ratio was like this? So it's very important. You don't every time have to work it out. If you know the ratios, you will be able to understand and answer the question. Then we must do incomplete dominance and codominance. Incomplete dominance is where we have a white flower cross a pink flower, or cross a red flower to give me a pink flower. That is incomplete dominance. Then codominance is where I have a white flower cross a red flower and it gives me a red and white flower, a flower with red and white color. Okay? Then the inheritance of sex inheritance. So it's XX cross with XY, that crossing. And then inheritance of hemophilia and color blindness and pedigree diagrams. Now, pedigree diagrams is going to be a session on its own, because it's usually about from 10 to 15 marks, and it's here where we expect the more or the higher difficult questions, the more difficult questions to pop up. And then lastly, we will do a genetic engineering. We will look at genetic engineering, what is genetic engineering and the role that it plays. I quickly want to come back to mutations. This was DBE 2015 paper. In there, the essay, because not a lot of question paper, this was for the June candidates. Now the June candidates, we call them senior certificate. Well, you are the national certificate, but it does not matter, they have the exact same syllabus. Right? It says, describe 2 types of mutations, how they lead to altered characteristics in the 4 genetic disorders that you have studied. Because it describes to you 4 genetic disorders in the examination guideline that you must expect. So describe 2 types of mutations is easy, it's level A. How they lead to altered characteristics, that is level C in the genetic orders. So if we quickly look at the memorandum, we say mutations refer to a sudden change in the genetic composition of an individual. So if you want to study something on mutations, I really suggest that you get hold of this paper because it was very nice there. What is the 2 types of mutations? We get a gene mutation, that is where the structure of a single gene change. That can be because the sequence change or whatever, so but it's the DNA in a single gene change. And the second one is chromosomal aberrations, that is where the chromosome or the number of chromosomes change. So when the number of chromosomes change, like in Down syndrome, it's called a chromosomal aberration. And then what is the effect of it can be harmful or lethal mutations, which can harmful or result in decrease of survival, and lethal will also decrease the level. Okay. Then what is the 4 genetic disorders that you must know? The first one is hemophilia. Now remember what was the question? It says, lead to alter the characteristic. So this is, you must work this out. This is higher cognitive level. So let's just see, how is hemophilia caused? It's caused by the mutation which lead that blood does not clot because the protein for blood clotting is not produced. What is color blindness is the next disease. What is color blindness? Person cannot differentiate between colors. Why? Because the necessary protein for photoreception is not formed. What is albinism? Albinism is the lack of pigment. Why? Because melanin is not formed. What is Down syndrome? Extra copy of chromosome 1 due to non-disjunction. So this is the 4 question. Very nice question, very nice summary of mutations and the 4 genetic disorders. Okay. I quickly want to come back to the genetic crosses. Okay. The first genetic crosses that before we do genetic crosses, let's just quickly look at whenever we do a genetic cross, there are some marks that you must get. Now that marks is now actually easy marks because you can get it without even doing the genetic cross. And the key ingredient for getting that marks is to follow the template. And it does not matter when you do it, you must always write the template. The template look like this. It says the phenotype of the first. You write the parents 1. What is the phenotype? The phenotype is what you see, nee? So it is, for example, black cross black or black cross white. It's the color you see. What is the genotype? Black can be big B, small b, cross big B, big B. Genotype is the genes that cause that phenotype. Then you must write the two processes, meiosis occur. So you have written the genotype there, so meiosis. Now you write out the gametes, or you can use the Punnett square. You write out the gametes, and you will get 1 mark for that. Then you must say the process of fertilization. That is the 4 marks that you get there, or the genotypes that you get here. So it's either there, or that you write out here, and the phenotype. So for P1, F1, or first generation, second generation, or first generation offspring, it's first generation offspring, you must get 1 mark. If you only write P1 and F1 with nothing in between, you will get 1 mark, because it's a given mark, it's a parcella given to you. And then the 2 processes, meiosis and fertilization, 1 mark, but it must be in order. Okay? So if we now look at genetic crosses, the first genetic crosses that I say we must look at is complete dominance. Nee? Now, complete dominance is where the dominant allele masks the effect of a recessive allele. Okay? Now, I say to you, if we look at complete dominance, we get 3 types of questions. Okay? So just remember, the big one will always show. Nee? It will always show that is complete. So we will get 3 types of crosses here, or 3 crosses here. The first one will be where the parents, the genotype of the parents is, let's say, let's use a big T and a small t cross. Okay, let's first do the 2 homologous ones. So I have big T cross small t cross small t. Okay? Let's say, and this T stands for tongue roller, that you can roll your tongue into a circle, or non-tongue roller. Non-tongue roller. Okay. So that was the phenotype, and this is the genotype. Okay, what must I then write? I must write meiosis occur. So what is followed there? It's the gametes. Okay, so meiosis, I will get T, and T. That's my gametes. Okay, what will my offspring look like? Okay, then we found fertilization. So I got my mark there for fertilization, and I got my mark there for, okay. So what will it be? I always prefer to work with a Punnett square. So I have big T, and here I have small t, small t. So that one will be T, small t, big T, small t, big T, small t. So my phenotype of my offspring, how will my phenotype of my offspring look? They will all be tongue rollers. Okay. And what will my genotype be? My genotype will be small t, t. And what will the ratio be? The ratio will be 100%. Okay. All the individuals will be heterozygous, 100% heterozygous. If I have a big T cross big T, small t. So I have a homozygous crossed with a heterozygous. Okay? Let's just see how my offspring will look like. T, big T, small t. So it will be big T, big T, big T, big T, small t there. So it will be, all of them will again be tongue rollers, the phenotype, but the genotype will be this one and this one, small t there. And what will my ratio be? It will be 50% to 50 or 1:1. So if I have a heterozygous times a homozygous, it's 1:1, 50 to 50. If I have homozygous cross homozygous, it's always 100%. Then the last possible one that it can be, if it's a heterozygous cross a heterozygous. Nee? And with this one, if I draw my Punnett square, it will look like this. I will get that one, and I will get that one, and I will get that one, and I will get that one. So how does my phenotype look like? Phenotype, I have 2 tongue rollers or 3 tongue rollers and 1 non-tongue roller, okay, for that one, okay. And how does my genotype look like? It's 1:2:1. So if we very shortly, what you must remember out of this is, remember I said to you, it's very important that you must remember the ratios in complete dominance. If we have heterozygous cross heterozygous, 100% of them will be heterozygous. That is the first one. The second one, if I have homozygous cross a heterozygous, can be this one also. It will always give me the ratio is 1:1 or 50%. 50:50. Okay? And the third one, if both of them are heterozygous, I will get 1:2:1 is my ratio there. So this will save you a lot of time if you know this. Okay. We will now look at examples. Okay, then the next type of question that they say we must get, that we must know, is they say that we must know incomplete dominance and codominance. Now, incomplete dominance and codominance is, the crosses look exactly the same. But you must read the cross, you must read the clue that is given to you. If it says to you the offspring is all one intermediate color, then you know it's incomplete dominance. Like, what is the intermediate color? It's like a red flower cross a white flower to give me pink offspring. So it's not red, it's not white, it's in between, intermediate color, it's in between, that's incomplete dominance. Now with incomplete dominance, the next thing, and with codominance, the next thing that you must remember is that it's 2 different alleles. Nee? So we don't work with big R, small r here, because both of them is color, but it's different ones. We use these symbols and we use these symbols. So again, look at the template. So we say there's red flowers cross white flowers. Nee? The red will look like this, the white will look like this. How will the gametes look? It will be R, or you can write it like this, it doesn't matter. And how will the genotype be? All of them will be this, nee? But the color will be pink, because somewhere in your clue it said to you it is an intermediate color or it is incomplete dominance. If we look at codominance, it's exactly, the crossing is exactly the same, but where's the difference? Look, even the genotype is the same as in incomplete dominance. Nee? The difference is in the phenotype. So with incomplete dominance and codominance, the only place where we differentiate, nee, is in the phenotype. In complete dominance, the phenotype is pink, and in complete dominance, the phenotype is white flowers with white and red flowers that is white with red spots, or flowers that is red with white spots. It doesn't matter. Okay, so this is the only way that we differentiate. And you as level 7 children must really, or learners must really understand that we use two different symbols, and that it's in incomplete and complete dominance. Okay. The next one that we must do, according to this exam guideline, is they say we must do the inheritance of sex. The inheritance of sex. Nee? So sex is determined, and this is very important because it looks like very simple, but sometimes the questions in pedigree diagrams come back to this cross. Okay? In sex, inheritance of sex or gender, nee? Sex or gender, there are two alleles, nee? that represent it. It is X and the allele Y. If you are a girl, you have the two alleles XX, and if you are a male, you are XY. Okay? So let's just draw the Punnett square here. So it's XY for the male there, and XX for the female here. So what is, that one will be XX, and this one will be XX. This one will be XY, XY. So what is my ratio? My ratio is 1:1. Okay? So there's 50% chance, although there's only one Y, there's a 50% chance that it can be a boy, or a 50% chance. Nee? So the ratio is 1:1. Easy cross, but sometimes it catch you right in there. Okay. Now the next one, which is a little bit more difficult, is the inheritance of sex-linked characteristics. Okay. Now sex-linked characteristic means it is gender related. It's gender linked. So what is the two alleles for gender? It's X and it's Y. Okay. But if you look at the appearance of the X and the Y chromosomes, then the X is usually a very big chromosome, while Y is a small little chromosome like that. So there's no space on Y. Nee? There's no space on the Y chromosome to carry different alleles. The only chromosome, a gender chromosome that carry alleles, is the X chromosome. So the moment you see sex link or gender link characteristics, you know it's carried on the X chromosome. You must never ever attach it to the Y chromosome. The Y chromosome is too small. The only thing it carries is the gender allele, or the allele that makes you a male. Okay? Because that is one of the biggest mistakes that you make. Okay? So let's quickly look at, if we have one of the diseases that they say we must study here is hemophilia. The other one is color blindness. Nee? So they say hemophilia is a sex-linked disease that occurs as a result of a recessive allele on the X chromosome. So how do we represent it? We have an X, and it is recessive, so it is a small one. So the other allele will be this one. So we attached it to the X chromosome. We don't say XX, we say XH. Where do we find it? We found it on the X chromosome, the female chromosome. If it's 8, it's a normal, the dominant allele. If it's small, it's the recessive allele. Right, now they say a normal father. Okay. So what is a normal father? How does a father look like? He have an X and he have a Y. but the disease is not carried, or the disorder is not carried on the Y. Now they say a normal father, so you know it's a big H. Nee? Okay, and a heterozygous mother. So what is a heterozygous mother? The mother have X and the X, and it's heterozygous, so one must be big and one must be small. Okay? And it says a heterozygous normal mother. Why does it say a normal mother? Because you cannot see, because it's dominance here. Because this one is big and that one is small, but you will see that the female is normal. You don't see the recessive allele. That is the principle of dominance. Nee? Okay, so let's quickly see what will happen in this cross. Okay, so we have, I always use a Punnett square, because the Punnett square is the best way to make sure that you don't have a mistake. Okay, so they say the father was normal, so I write the father in there. The mother was heterozygous, so she have a big one and she have a small one. So let's see what happens. I get X big H, X big H. So this is one of my children. And I get XH, big H, Y. So X, big H, Y. So this is a son, and this is a daughter. And because they have big ones, we say they are normal. It's a normal daughter. And it's a normal son. Okay? This one is look like this. So we say it looks like this, XH and XH. How will the phenotype look like? The phenotype will be normal daughter. Okay? This is the phenotype. But how will the genotype look like? In the genotype, it's not normal. She have a X, so she's known as a carrier. So the moment I ask you the genotype, it's a carrier. But the moment I ask you the phenotype, it's a normal. Okay, you say it's a normal daughter. And then the last one will be this one, XH and Y. What will be this one? There's nothing else. So this one says it will be affected. But we know it's for hemophiliac, so we will say it's a hemophiliac male. Okay, male. So very important. So what is the clue? What is important? Where do you lose your marks? Nee? It is when they ask you to differentiate between phenotype and genotype in sex-linked diseases. The other clue, it's only carried on the X and not on the Y. Okay, not on the Y. Okay, very shortly, I will go through them, and then the next one we will look at is dihybrid crosses. Okay, let's quickly look at some examples of questions. Okay, I also just took out some short questions here because it is very important that you see it is, ne? Okay, this one. That you see the ratio, how important it is. The fruit from an apple tree was harvested and sorted according to color. 94 apples were red, so it's 95. 97 were green, and 201 were found red with green patches. Nee? So red with green patches. What does this say to you? It says to you, it must be codominance. Nee? Because we see the two different characteristics. Okay. The next question. Okay, right, this is genetic modification. Homologous chromosomes, this is actually in meiosis, but homologous chromosomes are described, which one is the correct one, as being similar in structure and coding for the same characteristics, a product of division, identical daughter chromatids, 2 chromosomes that code, so it must be, it can't be A, it must be D, 2 chromosomes, because we are talking about a pair. Okay, homologous chromosomes, the moment I talk about homologous chromosomes, it is a pair. Okay, then they say 4 different phenotypes is in the F1 generation. Oh, okay, I see we've forgotten about the blood groups. Okay, so just before we answer that question, let's just quickly look at blood groups. Okay, when we look at the inheritance of blood groups, blood groups is an example of multiple alleles. That means that there are more than 2 alleles. Okay, there's an A allele, there's a B allele, and there's a O allele. So that is, there are 3 alleles. There are 3 alleles in blood inheritance. Okay. The correct way of using it, of describing it, is we say blood groups, blood groups, they use the symbol I. Okay. So if you are A, you will be homozygous blood group A like this. Or you can be homozygous this, because O is recessive. Okay? So that one we represent like this, and this one we represent like this. If you represent it by A and O and B, it is wrong. You must use it as IA, IB, or small I, small I. Okay? If you are blood group B, you can be homozygous, you will look like this, or you can be heterozygous, you will look like this. If you are blood group AB, you will be IA, IB. You can be nothing else. And if you are blood group O, it's small I, small I. So remember, in blood grouping, there are 3 alleles, of which O is the recessive one. So let's quickly look now at this question. 4 different phenotypes are possible in the F1 generation if the parents' blood groups are. So can B and B forms, because they say we want A, we want B, we want AB and O. So which phenotypes will give us this 4? B and B cannot be. Let's see A and B. Okay, if A is recessive and B is recessive, how will their children look like? Remember, I don't write F1, F2, but you must do it. So this is A and IB and I. So I will get A like that. I will get a B like that. I will get a A like that, and I will get a O. So I will get A, B, that's the blood group, AB, So I can get that one. This one that looks like this will give me blood group B. This one that looks like this will give me blood group A. And this one will give me blood group O. So 4 different phenotypes are possible if the parents are A and B. Okay, This one often crops up in here. So it says a father have blood group A. He has 4 children having the following: A, O, AB, and O. Okay? What will be the blood type of the mother of the above children? It's the same one that we just now. It must be A. The mother must be A blood group. Okay? Then 1.4, a tall pea plant was crossed repeatedly with a short pea plant. In these crosses, they only produced tall offspring. Okay? It is reasonable to complete. Remember I said the ratios. If we have big T, cross small t, all the offspring will be like that. So, the tall pea plant in the cross is homozygous. The tall pea plant is heterozygous. That's not true. The offspring are heterozygous for tallness. Okay? So that one is true. The short pea plant is heterozygous, not true. Here it said both the tall pea plant and the short pea plant. So the correct answer here is number C. Okay. In multiple alleles, remember what is multiple alleles? It's like blood group. So in multiple alleles, there are more than one gene. It's not genes. There are more than 2 different alleles. Nee? So this one is true. Nee? There are more than 2 different alleles. In a situation where characteristics is expressed more frequently in males than in females, nee? We can know it is sex-linked. So the alleles for the characteristics are located on the X chromosome. Because the male only have one, so if it's more frequent, it means that one of him have more than that. Okay? I just want to show you usually, section A, most of it, because it's so many marks, will be on genetics. So just make sure that you know your genetic terminology very nicely in there. Okay? Now, okay, I want to skip this one because it's almost diploid. Okay? There's another example. It says fur color in mice is controlled by a gene with 2 alleles. The homozygous mouse with black fur was crossed with a homozygous mouse with brown fur. All the offspring had black fur. Using the symbols B and big B and small b to represent the 2 alleles for fur color, show diametrically a genetic cross between a mouse that is heterozygous for fur color with a mouse with brown fur. Okay? Show the possible. So it says to you, you show a cross with a mouse that is heterozygous for black. So heterozygous for black will be big B, small b, with a mouse with brown fur, small b, because it said to you, you can now determine, it didn't say to you which one is recessive and which one is dominant, but that is what you as level 7 candidate must do. The clue was given in this part. It says to you, a homozygous mouse for black fur were crossed with a homozygous mouse with brown fur. And all the offspring had black fur. So that must tell you that black is this and brown is this, because all the offspring look like that and they were black fur. Okay? So if we just look at the memorandum there, okay, we just quickly look there, it says, just remove these pages, it says phenotype, so there's P1, F1, you will get a mark. meiosis fertilization, you will get a mark. Phenotype is black and brown. Genotype, black and brown. Nee, B, big B, small b, crown small b. Then we do the cross, and at the end there will be, the ratio will be 1:1. Okay, 1:1. Okay, so that is how we will do that cross. Okay. Now let's look at the next section. In an experiment to show codominance, okay, codominance, co means both colors appear. Cows with white fur, so my one symbol will be white, and bulls with red fur, so that's my symbol for red. Nee? So to be white, they have to look like this. Nee? A roan fur consists of patches of white and red fur. Roan cows and roan bulls were crossed, and it was shown above. Nee? So I got red cows, I got white cows, and I get red and white cows. Give the ratio to show the different phenotypes. So the ratio is 1:2:1, but it asked me phenotype. The genotype is 1:2:1. The phenotype is how they look. And all of this plus this will be red. Oh, no. I'm lying, sorry. It will be 1 red, 1 white (2 red and white. Okay? Explain why the cow and bulls of a genetic cross of RW will have roan fur and not red and only white fur. Nee? Because if the two parents look like this, the offspring will look like this. It will be red, and that one will be red and white. This one will be red and white, and this one will be white. So there will be white, there will be red, but there will be roan colors. Okay. So, and not only red and white. So it says there is red and white, but there's fur, because it is multiple alleles, or not multiple alleles, it is codominant alleles. Okay. The next question. Okay. Scientists, okay, that one is still coming. This one, the diagram below shows the alleles for height in a flower, in a color, in a flowering plant. So a moment you see this, what can I say about this alleles? This is heterozygous and this is homozygous. Nee? So T is for length, nee? And P is for purple. So this one is for tall and short, and purple and white. Nee? Now they say the plant is, so they want you to now describe the plant. This is a very good question, question D, very important, difficult question. Nee? So T is dominant, so what will it be? It will be a tall plant. Nee? And how will it look? It will be heterozygous tall. And what will the color be? Big P. So homozygous purple. So let's see which one is it. Homodominant for height, it's not true. Heterozygous for height, that one is true. And homozygous dominant for flower color. So number B. Right. Can you see? You must analyze, you must look at, you must break it down, and you must come up with an answer, a good level D question. Okay? This one, in humans, the alleles for short fingers, nee? If you have very short fingers, it's brachydactyly, is represented by B. Okay. Sorry, I want to skip that one because it's codominance. We will look at it just now. But let's look at this one, 2.3. Mr. and Mrs. Punelia are concerned that their baby girl does not appear to remember them. So they have a baby, but the baby does not look like them. They suspect that the baby were given to them in the hospital was not theirs. Mr. Punela's blood group is AB. So Mr. Punela is AB. So the phenotype look like this. And Mrs. Punela were B. So Mrs. Punela can be, so it's IA, IB. Mrs. Punela can either be this one. What is she? No, she's B, sorry. So she can either look like this or she can look like this. Okay. The baby they were given have O. Is it possible? How must you look to be O? You must be small I, small I. Okay? Okay, now let's look at the questions. Give the genotypes of Mr. Punela. The genotype is A, B, because he's AB. The baby girl, they say to you blood type I, so the genotype is small I, small I. Now they say to you, this is now higher cognitive, nee? This is now what you must be able to explain. Explain why the baby girl with blood type O cannot be Mr. and Mrs. Punela's daughter. Why can it not be their daughter? Because now you must say Mrs. Punela can either be heterozygous or homozygous. That's the first mark. She can either be homozygous or heterozygous or homozygous. The next mark is to be O. You need small I, small I. She cannot inherit a small I from her father because he's A, B. And if her mother is like this, she can only inherit one. So there's no way that this baby can be Mr. and Mrs. Punela's baby. Okay, so that is very high cognitive. Analyze the situation, look for the relevant information, get to an answer. Okay, can you see? No? Then explain why the use of blood types for parental testing is not conclusive. Okay. In the olden days, they have used blood types for parental. But a blood type, many males can have O blood types, and many males can have AB. And if the baby is AB, then it's possible that it can be that group of men, but it does not pinpoint the specific male. Okay? And use your knowledge of sex chromosomes and explain why. Explain. It says do not use a genetic cross. It says explain. So although this is a level B question, an easy question, many learners got it wrong because they have not written it out. They have actually done the genetic cross. So if we just see why does the Y, look at the question, use your knowledge of sex chromosomes and explain why the sex of a child is determined by the male gamete. So you start to say, normal females have 2 chromosomes. Normal males have 1 X and 1 Y chromosome. The female always provides an X. Always, because she has XX. So she always gives an X. And the male, and if it's fertilized by another X, it's a daughter. And if it's fertilized by a Y, it's a boy. Okay? So that is why the male gametes. If the male contributor X, it's a daughter. If the male contributor Y, it's a son. Okay, let's look at the next question. A farmer has an orchard of apple trees. Each apple expressed red and yellow color. So what does it tell you? Red and yellow. So both are there, so I know this is codominance. Ne? To extend his apple orchard, the farmer collected seeds from the red and yellow apples and grew them. When the new trees matured, he found that some trees produce red apples while others produce yellow apples. Ne? Because the parents look like this. Ne? They were red and they were yellow. And all the offspring they produced were trees that are red with yellow patches. Ne? So do a cross to show this F1 generation. Okay, so at the end it is, he says, when the trees mature, right, use a genetic cross to explain his results in the F1 generation. Okay, so you have an orchard of trees, ne? And all the trees, all the ones that he got was red and yellow. So he crossed, so he used red and yellow as the parents, and he crossed them, and at the end he got red apples, red and yellow apples, and yellow apples. Okay. So where does the difference lie? The difference lies in the phenotype, ne? Because this is red and yellow apples. Okay, so that's the first question. What proportion of the F1 generation will be red, yellow apples? It will be 1:2:1, so it will be 50%. Okay. The farmer saw, this is a very, very good level C question. The farmer saw that red, yellow apple sells the best. Name the phenotype of the tree that he should cross in future in order to assure that all new trees will produce only red and white apples. Okay. So how will what do we want? He wants red and yellow apples. So how must the parents be? If he only wants red and yellow, it means the parents must be homozygous, yellow, homozygous for red and homozygous for yellow. So the offspring will all be heterozygous or red and yellow. Okay. Different application, good example of a level C question. Okay. Then we often, like I said to you, in all the papers, there will be an investigation. Okay, so this one is not an investigation in terms that it asks you the conclusion and the hypothesis, but let's read here. Learners want to investigate eye color in fruit flies. Red eye color is dominant, while white color is recessive. Okay, so they tell you the symbols is big R, small r. Male fruit flies homozygous for red eye color. So it's big R, ne? Were bred with female fruit flies homozygous for white. So this was the original parents, ne? Okay. Show the possible phenotypes and genotypes of the F1 generation. So you will do your genetic cross, you will write it out neatly. Okay, so let's just see, you will say red, 1 mark for phenotype, 1 mark for parents and offspring, 1 mark for the 2 processes. The phenotype, red cross white, 1 mark. 1 mark for the genotype, homozygous red and homozygous white. Ne? And at the end it gives me 100% red. Okay, straightforward one, not a difficult one, but if you didn't know um, um, recessive and dominant, right? Let's look at this one. This one is altogether a level C to D question, because what you have done in the class with sex link, okay, is you have done either hemophilia or blindness, color blindness. Now let's look here. It says, a human brown tooth enamel is inherited as a sex-linked recessive trait. So sex-linked immediately tells you it is on the X chromosome. Okay? Now it says to you, and it's recessive, so it must be small. A man with brown teeth enamel, okay? So it means the man is X, Y, and is recessive, so he will have a small one there. Nee? Marries a woman with white teeth, but whose father had brown tooth enamel. So what does it tell you? It tells you the mother will be probably be heterozygous. Use XB for white teeth, so it's XB, because we know she have white teeth, and XB, so we know it's the father look like this, ne? And her father was brown, so she will be X with a small one. Explain why the woman is heterozygous for tooth enamel color. Now you must look very carefully to the clues that were given to you. It says you must have the recessive allele for brown tooth color. Okay? And it says to you the woman had a father with brown. So the woman's father looked like this, right? He looked like this. So the only X she could inherit from him was this one. And then she's normal, she have white tooth. Use a genetic cross to show the possible offspring of their children. Ne? So it means you will use that crossing, you will use the father, if brown teeth enamel, the mother have normal teeth enamel, but because her father had brown, she inherits that one, so that's why she looks like that, and then it's just a normal genetic cross. Okay. Okay, so I hope that I'm going to end this session here. I hope that you understand now that what is the different types of crosses that we must know. Just want to get that one page. Okay. If we go back to the different crosses, it says we must know complete dominance. Incomplete dominance, we can get 3 types of dominant crosses. We can get homozygous, cross homozygous, then the offspring is 100% heterozygous. We can get homozygous, cross heterozygous, then we get a 50:50. And homozygous, heterozygous, cross heterozygous will lead to a ratio of 1:2:1. So you must know that 3 crosses. Then we looked at incomplete and codominance. Same crossing, the only difference is in the phenotype of the offspring. Inheritance of sex, XX and XY. Inheritance of hemophilia or blindness, where you know the mother, it's carried on the X, and the father only have one X, so depending whether it's heterozygous or homozygous or recessive, you will put it in there. Okay, we will meet again. Thank you. you.